currently and has been at the University of Idaho in Moscow for some time, following training at Michigan State. Larry is an ecologist, a microbial ecologist, and interested in many of the basic themes in microbial ecology, has focused his efforts in a couple of areas, one of which he's going to tell us about this afternoon concerning the human vaginal microbiome. And we're delighted you can be here, Larry.
Well, I very much appreciate the invitation to come and tell you about some work. I found that it was a difficult challenge, actually, to put together a talk for what I thought was a rather diverse audience. What I can tell you is I'm going to avoid getting into the real grimy details of anything that has to do with characterizing the vaginal microbiome and stay away from all the bioinformatics and all the rest of it. Instead, I'm going to take a rather high-level view and be a little bit outrageous. I had a very good plan when I left home yesterday, and then I was delayed in Salt Lake City, and I got 3 hours of sleep. And so, as Karen said, it's now all poorly formulated in my brain. But we'll see if I can pull it out and tell you some of the things about this that you might find interesting. One of the things I'd like to do is, as an outsider to this field, meaning the field of human microbiome, there have been some facts and information that has been in the literature. These are stories that have become a part of the folklore of the vaginal microbiome, and I would like to, as I've sort of questioned them a bit and ask whether or not we actually have the data to support the statements that are being made. And many times I find that it's lacking. And I was talking to Brendan during the break, and I said, maybe it's a function of getting older, but I feel a bit like a curmudgeon. I can say whatever I want, and I have nothing to lose. And so I'm going to do that a little bit. I'm going to take some liberty and point out some things that may not be true, but are certainly worth thinking about and deserve a little bit closer examination. The title of my talk is As You See It. The subjects I'm going to cover are the common wisdom of the vaginal microbiome, which are the stories that are told about the vaginal microbiome. I'm going to talk about differences that occur among women in terms of their community composition and the dynamics of the vaginal communities over time. Talk a little bit about community stability, the concepts of resistance and resilience, and how that may be important to whether or not the community enters a disturbed state. Because in a disturbed state, many ecosystems are more subject to invasion. That's what the ecologists call it. Infectious disease doctors call it infection. And so we'll talk about that and how the factors of resource competition may influence the dynamics of the system and how that might govern whether or not a system actually becomes destabilized and subject to invasion. And the last thing I'm going to touch on a little bit is some of the work we're doing on community genetics, which includes to do comparative genomics of some of the major players in the community in an effort to try to put together an ecological network of the interactions that occur among the species and with the host. And many of the studies seem to be pretty important. As you'll see, the resources may become a pretty important driver in this system, we think. So this is some of the common wisdom, and this is a story that really began a long time ago when this German physician first described the bacteria that were found in the vagina. That was called the Doderlein bacillus, and later it was called Lactobacillus acidophilus, and who knows what it's called now. But this physician was the first one to describe it. And in the subsequent 100 years, there was a story that came out that said that lactobacillus was characteristic of normal, healthy, reproductive age women. These are the bacteria that you would find. This story right here is true, in that in many healthy reproductive age women, these are the dominant bacteria that are present. The problem with this definition is that the reverse is not necessarily true, and that the absence of lactobacillus is not necessarily to be equated with disease, and yet it has been talked about in that way over the years. And it's thought that these lactobacilli play a pretty important role in preventing the invasion by non-indigenous organisms, so pathogens being foremost among those, and that occurs through the production of an acidic environment. And it's thought, and it's talked about, that if the pH of the vagina is less than 4.5, you're healthy, and if you're more than 4.5, you're not healthy, and that it's a very simple mechanism of exclusion, which is really a pH environment. That has a lot of appeal because people are familiar with fermented foods and the idea of fermentation as a way to preserve foods and to exclude non-indigenous organisms that might spoil it. And it's sort of caught on. It's very popular and talked about quite a bit, as though that's exactly what had happened. And this, of course, the lactic acid is the fermentation product of the lactobacillus, and they grow on substrates produced from the host, produce lactic acid, reduce the vaginal pH, and voila, we're all set. In the vaginal microbiome, this is a really remarkable environment because it's subject to perturbations of various kinds. Some of them are from the host. Menses in reproductive age women would be a good example of that. Other kinds of disturbances you can imagine might occur during sexual intercourse or other sexual practices. But it seems to be very resilient. It comes back in a very remarkable way most of the time. And even though people want to focus on disease because it's, I guess it's interesting to talk about, I'm struck by the reverse of that, which is most women are healthy most of the time. And that means that this system has co-evolved with the host since the human population was founded to be very, very effective at preventing infection. As an open orifice that is essential to preservation of the species, because it's reproductive, it has to work well. And the fact that it works well is evidenced by the fact that we're in this room. Otherwise, we'd be extinct. So these are the events that occurred. Also, a succession that occurs during life. During birth, it's thought to be sterile, but during the birth process, if it's a vaginal delivery, it's thought to be colonization by the bacteria from the mother's vagina. During puberty, there's subsequent metabolism of the estrogen that is carried on in the newborn infant. Once that's dissipated, then you have colonization by enteric bacteria and anaerobic bacteria, which haven't been very well characterized, and that continues until puberty, so about age 11 or 12, and then you have an increase in the level of estrogen, which is thought to then precipitate a change, a selection for the lactobacilli, which can accumulate and metabolize the estrogen, which is in the vaginal epithelium. So there's a thickening of the vaginal epithelium, an accumulation of glycogen, and now you have the lactobacilli that show up that are characteristic of women in their reproductive years. During menopause, there's a reversal of what happens during adolescence. And I don't know if that's exactly true, but it seems like it's true. There's a reduction in the level of estrogen, there's a thinning of the vaginal epithelium, and the composition of the community tends to change back to what you see during puberty. And so it's really driven by estrogen levels, hormone levels, seems to be driving the selection of the flora that are found in reproductive age women. The bad news for many of us is that most of the studies have really focused on reproductive age women. And there's very little known about women that are in puberty or in menopause. I mean, if you put this on a scale, I would guess there's probably 100 times more papers, 1,000 times more papers when it comes to adolescence, and maybe 100 times more papers relative to what's known in menopause. So there's large segments of the population. When we talk about the vaginal microbiome, we're really talking about the vaginal microbiome of reproductive age women. And please forgive me, but that's the way it is, and we're working to change that a bit. This is something Mark Twain said, and I think it's very true. There are things that are told in introductions, to papers, in review articles, in textbooks, in the hallways, and it sounds true, and it could be true, but the problem is it's not true. And then you walk around thinking that you know something when in fact you don't, and when you go dig to see, well, what's the data to support this? You find out that it's a little bit missing. It's a little thinner than what you'd like it to be such an important fact, especially when you're making diagnoses about health. You wish that the data was a bit more robust. So here's one that I really like. Women are more or less the same. They have more or less the same bacteria in their vaginal microbiome, and that these communities are more or less stable with the possible exception of menses, which everyone says, yeah, but that's menses, and then as soon as that's over, it's reestablished. And so women are more or less the same, and the communities are more or less stable. But we've published a couple of papers in recent years that sort of turn these things upside down. It turns out that women aren't more or less the same, and they aren't more or less stable, and so this all sort of falls apart. And women are, and their vaginal microbiota are, and that's about what we know. So we're really starting out from a pretty basic level, trying to reestablish a foundation of knowledge so that we can then make better informed diagnoses of disease and understand how to maintain health, and a number of things which we're all very interested in. So this is one study I'm going to quickly go over. It is a cross-sectional study that was published in PNAS with my collaborator Jacques Revel. Most of this work I'm going to talk about is done with Jacques Revel, who's at the Institute for Genome Sciences at the University of Maryland School of Medicine. This is a study in which 396 women that represent 4 different ethnic groups were sampled in a cross-sectional manner. There were 100 blacks, whites, Asian, and Hispanic women, and the composition of their communities were determined after they had self-collected a vaginal swab. We'd extracted the genomic DNA, amplified the 16S genes, and sequenced them using bipyro sequencing. So this is the usual complicated heat map that people like to show about these things. I don't know why they do that, but they do. Basically, what you have on the left-hand side are the list of all the taxes that are present. The 396 women are shown on the dendrogram that's up above. The different colors represent the relative abundances of different populations, and what you can see is that the composition of these communities can actually be clustered into 5 groups. Now it's getting simpler. You just look at the top bar. You have group 2, group 3, group 1, group 5, and group 4. I don't know why we didn't number those sequentially. It would have been easier, but that's the way it turned out, and now we're stuck with it. What you can see here is that group 2 is really characterized by the predominance of an organism called Lactobacillus gasseri. Group number 3 is dominated by Lactobacillus iners. Group number 1 is crispatus. Group number 5 is Lactobacillus gasseri. And group number 4 has a large number of other organisms, notably absent are the lactobacilli. And these women were, in fact, symptom-free, and we believe them to be healthy. And so this is an example of where, well, what about this group over here, which doesn't follow that rule that Dr. Dodenline wanted us to remember. Before we get any further, these are the community types that actually represents a pretty large study of 400 women. But because of the way the sampling was done, this represents 95% of the women in North America and these 4 ethnic groups that we would sample. But it means that there's 5% we don't know anything about, except that they aren't any of these. There's something different. That's 1 in 20, which for a clinician might be important to remember, that somebody's going to be, every 20th woman is going to be different than everybody else you know about, and so that might be worth remembering. And we call these community state types, and you'll see why that's important in a minute. So each composition at each point in time is a community state, because it's going to change over time. And we do think that the function of these communities might be conserved because the organisms that are over here in the red, which includes Streptococcus and Megasphaera, they all produce lactic acid as a fermentation product. And so it could be that the function of these communities is conserved even though the species composition varies. And so the importance of lactic acid, or at least the question of what is lactic acid doing, could be true for all of the communities that we're looking at. One of the things that was important was that among the ethnic groups that we looked at, the 4 ethnic groups, they all had the same 5 kinds of communities, but they differed in their relative abundance, or their proportion in the women. So if we start out with the white women as an example, this group right here, the big green sector on that pie chart, are the communities that are dominated by Lactobacillus crispatus. And that is something that in the literature, Lactobacillus crispatus and vaginal communities have almost become synonymous. And what you can see is that as you go from the white women and the Asian women over here to the black and Hispanic women, the relative abundances of the green pie sector becomes a little bit less. And what increases are the group 3 and the group 4, especially this one, because the group 4 is the one that's not lactobacillus dominated. It's the one that was on this side of that heat map. So that could be important. If you believe that lactobacilli are important to health, then you might begin to wonder about the health of women that are Hispanic or the health of women that are black. And this could have important consequences for the epidemiology of different diseases, which we could talk about if time permits at the end. One of the things that was important was that among the ethnic groups that we looked at, the 4 ethnic groups, they all had the same 5 kinds of communities, but they differed in their relative abundance, or their proportion in the women. So if we start out with the white women as an example, this group right here, the big green sector on that pie chart, are the communities that are dominated by Lactobacillus crispatus. And that is something that in the literature, Lactobacillus crispatus and vaginal communities have almost become synonymous. And what you can see is that as you go from the white women and the Asian women over here to the black and Hispanic women, the relative abundances of the green pie sector becomes a little bit less. And what increases are the group 3 and the group 4, especially this one, because the group 4 is the one that's not lactobacillus dominated. It's the one that was on this side of that of that heat map. So that could be important. If you believe that lactobacilli are important to health, then you might begin to wonder about the health of women that are Hispanic or the health of women that are black. And this could have important consequences for the epidemiology of different diseases, which we could talk about if time permits at the end. This matters a lot, right? Because when a woman goes for an exam, she can be told she has a normal pH. She can be told she has a normal vaginal odor. She can be told she has a normal discharge. Normal is a mathematical abstraction. We made this up, right? The calculation of normal. And yet, when you're labeled as normal, you can feel pretty good about yourself. The problem is that if you choose a different word, like you're not average, or you're atypical, or you're strange, or you're odd, or you're unusual, or you're abnormal, or you're weird, or you're different, or you're peculiar, it has the impact of having women's self-esteem being determined by the words that are used to define a very intimate part of their body. And so the whole idea of autonomy and respect for individuality falls apart. And so if you don't buy into personalized medicine, maybe you can buy into treating people as individuals. Like I said, I can really get going. I hadn't put those two slides in. So where did we get this idea that vaginal pH of 4.5 was so important? I mean, it does take a continuous variable, which is pH, and make it categorical. If you're above pH 4.5, you might not be healthy, and if you're below pH 4.5, you are healthy. It never comes out, you don't really say it that way. What you say is, and the CDC says it this way, an elevated pH is common with bacterial vaginosis or trichomoniasis. So it says that if you have a higher pH, you may have a problem. Here, it comes down the same way. The Human Microbiome Project set as an exclusion criterion a pH of 4.5. If a woman wanted to enroll in the project and the vaginal pH was 4.5, she was not included in the study because the purpose of the study was to study healthy women. And this keeps showing up over and over again, countless review articles and commercial over-the-counter kits that measure vaginal pH as if it were a bright line. And yet we have to wonder whether or not this is true or not. So if we assume that pH is in fact a continuous variable, then we can begin to make little graphs like this if we assume it's normally distributed. And what it really says is that 14.3 million women are going to be in the tails of that distribution. Some are going to have a very low pH, some are going to have a very high pH, and yet all could be normal and healthy. So as long as we're into normal and healthy, these are two words that haunt me. Every time I try to get a grant proposal reviewed or submit a paper, people say, did you sample normal and healthy women? And we say, well, we think so. They said they were. They went through on a clinical exam. Everything seems to be okay. But I just went over the concept of normal and how that's a problem. So now let's go to the problem of healthy. Well, this is the World Health Organization definition of health. Health is a state of complete physical, mental, and social well-being and not merely the absence of disease or infirmity. So I've decided we're all not healthy, okay? We're just in different stages of decay. And so this is really an issue because you have to understand that when we're studying individuals in the human microbiome, we really are not necessarily studying healthy and not healthy. We're studying people that have different degrees of health or differential risk to disease. But there isn't some kind of an ideal form, as Plato would have us believe, that we're all being compared to, that this is normal and healthy, and we know what it is, and we can just describe you in relevance in comparison to that ideal form. Right? That's not what we're really doing. We're trying to describe the variation that occurs over time. Otherwise, you might be like the old Texas joke, which I'll tell you if you want to hear it later. So you're healthy, you're not healthy, you're healthy, you're not healthy. Because it's going to change over time as your microbiome changes. So we also went back and questioned a few things. So this idea that pH 4.5 was magic, and if you just get below pH 4.5, then you won't have organisms grow, that these non-indigenous organisms won't grow at less than 4.5. Well, I knew that that really wasn't true from work I did, even with my hands, a long time ago. And so recently we just redid it again. We took Enterococcus faecalis, we put it into media, we buffered it with MOPS and MES buffers so we would keep the pH, and we just could test it whether they could grow. And sure enough, they don't grow at pH 4, but at pH 4.5 they do grow. And in fact, if you look at the rate of growth, you see it's almost the same as at higher levels. It's about half or a third as fast. But the idea that they can't grow in an environment at pH 4.5 is clearly not true. It's also not true for E. coli. Maybe we'll do some more growth studies like this. But it's a very simple test that says that paradigm, that rule that gets repeated so often may not be true. So it's not that simple that you simply have an acidic environment. Okay, do these communities change over time? This is one where we wanted to know whether or not they could change over time and whether or not there were temporal differences and whether or not we could identify women that were at risk, because these disturbances or perturbations could be important. This is the pattern that you might expect. This is a rather complicated graph, but in the top panel is a heat map again, where these are the taxa that are shown. This is an interpolated graphic of exactly the same things, so you can see changes in community composition over time. And these are a lot of metadata on sexual activities, behaviors, and practices. The only thing you need to pay attention to are the little red dots, because that's menses. So every time this woman had her menstrual period, there was a shift in the composition of the community, and it occurred every time that she had menses. Interestingly, the change was exactly the same. It went from an L. crispatus dominated community to an L. iners dominated community, and it was very regular. This was a study of 32 women that were sampled over 16 weeks, where they were sampled twice weekly. Okay, and so we have a temporal data, and this is exactly what people, if you were to guess, right, a little bar bet, what would it look like? This is what you might expect. Changes during menses that then returns to normal. Of the 32 women that we looked at, this is the only woman that followed this pattern. Other women had this kind of a pattern, where this was her menstrual period. It's a bit shorter. In the first one, there was almost no change whatsoever, and even over here. But here, there were two massive changes. The two massive changes are different in terms of the kinds of changes that took place. You can tell by the differences in the colors that took place. And so this is very different, and the change is different in terms of not simply a switching from one lactobacillus species to another. So you get this kind of a pattern. There are also women that don't change at all during menses, or you can have women that do this kind of thing. So here's one on the left, where the community is very, very dynamic on the first half of the study, and then it undergoes a transition to what would appear to be an alternative equilibrium state. So the right side of this graph doesn't look like the left side. There's been a change. There are no spikes that occur during menses that are nicely correlated with that, so it's different than what we saw before. But there is suggestion of an alternative equilibrium state that exists, and it can look like this as well. And if you want to, it can look like this, and it can look like that. And you can see that these are very dynamic systems that change over time. We have something I'm going to breeze through very quickly. You can do this community composition to determine the community state. And if you do that on consecutive days or twice a week or so on, you can string these together and begin to see patterns of, of this, where I was going to point this screen. Where this is the way we would classify the first half of this, and then this is the way we classify the second half, and it has to do with the composition of the communities, and it's easier to see than the patterns when you look at all the 32 women lined up together. No two women are alike. That's the take-home from this. They have different kinds of transitions. Some are more stable than others. Some are less stable. Some vary, with a regular periodicity. Sometimes it's just a one-off in the middle of their of the pattern and so on. So these systems are dynamic, and you have to ask yourself what's going on in these communities and what's causing them. This is a movie that some of you have seen, but we've taken these 5 different community states and we've plotted them in a PCA plot, a tetrahedron, where these represent in the corners the different kinds of communities. There's one in the middle of this tetrahedron you'll see in a moment. And so each dot represents a single woman from the cross-sectional study, which I told you about, the 396 women. The large yellow ball right here is this woman over here. And we're going to see how her community changes over time. And it's a little bit more interesting than a heat map, because you can see that she initially starts out sort of here in the middle, then she decides that she wants to go down here in the red corner, which is dominated by Lactobacillus crispatus. After a period of time, she shifts over to the green corner, which is Lactobacillus iners. And then she's going to go back towards crispatus. And then she's going to end up back in the middle of the tetrahedron. This is over a 16-week period. And so when you say, what is the vaginal community of a woman, it's going to have to be in, at what point in time would you like to talk about. This has important implications, because if these different states differ in their risk to invasion or risk to disease, then defining a risk to disease for this woman has to be refined to time. In other words, her risk will vary depending upon what day it is and what the events are that are driving the changes that we're seeing, of which we have absolutely no idea what's causing these changes. But we do have some alternative hypotheses. There's nothing remarkably surprising about these, the dynamic equilibrium, meaning that it sort of sits there, and so I think of it as vibrating in space. The community space hypothesis says that you can look at one face of that tetrahedron, and the woman's community can move across that face, but can't access all of the genetic space that's represented by the tetrahedron. It's limited to what can be done. The alternative equilibrium hypothesis is another example where there's a flip from one state to another. And the community resilience hypothesis, which is what I call the rubber band hypothesis, that the community can change in response to a disturbance and then comes back to a ground state. Face level, surface level analysis of just looking at the plot say that all four of these examples exist in the set that we've looked at. And we're deep in the middle of looking at 135 women that were sampled daily, and the results are very similar, and it appears as though there are different rules for different women on what seems to be going on. So recap. The function of lactic acid is apparently conserved. These communities can change markedly. They need to be accounted for in determining risk, and we need to reevaluate and use carefully the words healthy and normal. So the stability of these systems becomes important because of the idea of a weedy species. From plant ecology, you can come up with very simple statements that say that if two plant communities differ in terms of the species of plants that are present, they may differ in their ability to and how they respond to disturbances. So this would certainly, if we just take that idea and put it over onto the vaginal microbiome, these will have different abilities to respond to disturbances. In one case, the disturbed state itself could elicit signs and symptoms of disease, such as in bacterial vaginosis. In another case, as I've already mentioned, it could result in invasion by weedy species, which other people would call infectious organisms. And so these are the reasons why we need to be thinking about factors that affect the stability. Bacterial vaginosis is a very important condition that can affect, and the prevalence is about 28% in North America. It can be higher if you go into different populations of women. And it's important because of something I'll show you in a minute, and that's it increases risk to lots of different things, sexually transmitted infections, preterm birth, a lot of issues. Look at the description from the Centers for Disease Control. The normal balance of bacteria in the vagina is disrupted and replaced by an overgrowth of certain bacteria. That's pretty specific, right? I mean, that says that something's going on, it's all falling apart, but we don't know what's causing it. Sometimes it clears up without treatment. Other times it needs to be treated with broad-spectrum antibiotics, usually metronidazole is the case. So it's common and it has adverse effects. And it's the curse of postulates. The world is out there trying to find the causative agent of bacterial vaginosis, when in fact there's pretty good evidence that it's an ecological problem, that it's a system that is disturbed, it's a disturbed ecosystem that can persist for long periods of time. And the sooner we get to where we wrap our heads around an idea and we stop searching for a single organism as the causative agent, the better off we'll be. I'm going to quickly go through one of the things, community genetics. There's one part I wanted to get to because it gives you an idea of where we're going in this field. Resources are important in the vagina, and unlike many other microbiome sites that are studied, all the resources are provided by the host. So this is unusual. There are no creams, no lotions, no food, no anything. It's all provided by the host. So you can guess, and guess correctly, that the host is going to have a big influence on what bacteria are present, what they're doing, how they're doing it, and so on. So it's going to be a complex set of resources that are provided to the bacteria, and they're going to determine the complexity of the dynamics that interact between them. The reason why that's important, and I'm going to skip to this, is that these lactobacilli are known for their nutritional requirements. You have to provide a lot of things to grow them in nature. And most of the food lactobacilli have genomes that are on the order of 2.5 to 3 million base pairs. If you look at the lower level, the lactobacillus inert, this is 1.2 to 1.3. It has an ameliorated genome. It makes you want to think about becoming an obligate endosymbiote of the host. These 4 species of lactobacillus are only found in the human vagina, so far as I know. And so it's an example of really a coevolution of these lactobacilli with the host. And so the sharing of the nutrients could become pretty important. So where are we going now? Well, we really don't understand what functions are being performed by the microbial communities that are important to maintaining health. I personally am feeling less good about the idea of lactic acid and pH. And we don't understand the ecological interactions. Treating this as a true symbiosis, if you will, between the bacteria and the host, it could be important. Certainly the host is benefiting from the presence of the bacteria, and the bacteria are entirely dependent upon the host for their nutrients. And so this is a classical symbiotic relationship. And this big question down here is, if lactobacilli aren't present, then how are these women possibly remaining healthy? So there we are. We have a mutualism, and it has multiple components. And the sooner the people in the microbiome field start thinking more about the host, women are going to be the beneficiaries. And with that, I'll stop and recognize, as I mentioned earlier, Jacques Ravel is a close collaborator of mine on these studies, and we have a team, a cast of thousands, that work on this. And I'll stop and take any questions if there's still a moment.
Thanks, Larry. That was great. Yeah, we can take maybe one or two questions, and we'll hold the rest and have a group discussion in a bit. Claire, did you want to?
Yeah, I have several questions. I'll just ask one now and save the rest for the discussion this afternoon. Based on what little I know about ecology, I'm under the impression that community diversity or ecosystem diversity is a good thing. Do you have, I'm sure you've looked at how these different communities compare in terms of sort of standard measures of diversity. Are they similar, or are there differences? Does that really matter, or am I totally off base?
I don't think you're totally off base. It is generally, there's a lot of debate about diversity and stability indices, and generally it's thought that as communities are more diverse, then they're more stable. But we have not actually done that analysis to see whether or not there was an increase in alpha diversity, meaning the number of taxa that are present, and the stability. There is, however, in this, and we're guilty of this, we call the one that has greater evenness, meaning we detect larger numbers, we call that more diverse, and that's really not true. It's more even. We don't know the full extent of diversity. But that's a great question. It could be really important to understand, well, if there's something that's reducing diversity and making it more fragile.
Quick question?
Sure.
Okay.
Interesting on the variability between women. Did you look at all, since it's a mucosal associated microbiota, did you look at secretor status or blood type?
No.
Given that there might be a glycan influence
No.
in the variability you see?
We haven't. We should. We're guilty.
Thanks a lot, Larry. There are more questions that we'd love to return to, so if you can all just sort of hold them, and we will come back to the group discussion.